This is the third video in a sequence of videos looking at titanium oxide. The example I'm going to use here is an experiment in which a titanium foil had the oxide and any contamination removed using an ion gun. So the ion gun has implanted argon, which is typical of these two peaks here. This is the argon 2p, and what is left is minimal amounts of oxygen, small peak here, and there's no evidence, certainly in this survey spectrum, of carbon. So the surface that we start with is relatively clean. And then a series of measurements are performed in which an argon cluster source with 1,000 atoms in the cluster is used to maintain the surface in a relatively clean state with respect to the carbon. The oxygen, however, continues to grow. The ion beam is not sufficiently powerful to remove the oxygen, but it does maintain a surface that does not have any hydrocarbon or CO type bonds. Only carbon that is possibly bonded with the titanium is left on the surface. And the idea of the experiment is to observe changes occur within the titanium 2p as a result of the oxidation sequence. So we can see that the surface is oxidized with time. You can see the color coding that applies to these vamous blocks indicates that we started off with the metal and it progressively oxidizes to produce a shape in the data which is very different from the original metal spectrum. In order to gain an understanding of the titanium 2p, we're going to use several options on this lower toolbar, and these include the PCA toolbar button. This will help us to assess the number of different shapes that are significant to these data. It will also provide the basis for choosing the number of abstract factors that we calculate from PCA to reproduce the data. The idea there being that we enhance signal and suppress noise by only using abstract factors that include signal, and they may also include some noise, but if we can identify all the abstract factors that are definitely noise and exclude them from the calculation, we will reduce the signal to noise problem within data. The next option, once we've improved the signal to noise in data, is we'll apply the difference operation, which will allow us to take two spectra and then construct a new VAMAS file that contains spectra that represent different proportions of the two selected spectra. And the different proportions will show us shapes that would not be in the data set but might be interesting in terms of interpreting the data. And once we've come up with shapes that we think do interpret the data, we'll use the linear analysis button to calculate a linear least squares solution based on the component spectra that we've just calculated, because they will be of no value if they don't actually reproduce the data in terms of a linear least squares fit. So we're going to put together these options by considering not just the titanium 2p, but we're also going to include the oxygen 1s, because we can interpret the titanium 2p better if we watch what happens to the oxygen at the same time as we allow changes to occur using the difference option between the titanium peaks. Since I want to combine the oxygen 1s and the titanium 2p, the first thing I need to do is construct a new data file that contains just these two pieces of information. And they must be in a form that I can then apply PCA, etc. to analyze the data. And the way you do this is to make a selection of all of the VAMAS blocks that are of interest, then display in the left hand side display tiles that contain the two spectra of interest on a row by row basis, and that's achieved using tile by row toolbar button. So make a selection and then apply this button, and we end up with a scroll list that contains all of these spectra arranged with the oxygen and the titanium according to the experimental variable. So having prepared the scroll list in this way, we're then able to construct a new file using another toolbar button that says merge irregular. So each one of these display tiles with the oxygen and the titanium correlated in this way will form a new VAMAS block in a new file. So I've now got all of the data organized in VAMAS blocks containing oxygen and titanium for each of these experimental variables, so we can now process these data and see what shapes emerge from the spectra themselves, rather than guessing at what we think ought to be there. We can find out how many different shapes we might expect to see in these data if we simply apply PCA to these data. And then the abstract factors that are calculated are an indication of the signal that's required to reproduce the entire data set. And when I say reproduce the entire data set, what that means is that we have abstract factors. They're not necessarily spectroscopic in significance, but they are mathematically significant to the data itself. So if we were to take the abstract factors that just have signal, that's obvious signal, more signal here, until we ultimately reach a point where it looks mostly like noise. So if we only take the abstract factors, so all of these above this point, they appear to have some shapes of significance to the signal, not just noise. We don't need this abstract factor to reproduce the data. All we would be doing if we included this abstract factor into the calculation would be to add back noise. So the idea is to avoid adding the noise that is actually within all of the rest of these abstract factors. And so if we only include in a least squares fit a set of abstract factors where we see something that might be important, then what we will do is we will have better signal to noise, and the noise will be suppressed in the rest of the data set. So that's the basis of how you might use PCA and the reproduction of the data with PCA to improve the signal to noise in your data set as a whole. And this is important when we come to do difference calculations, because when you do a difference calculation, the difference between noisy data is amplifying the noise. So we really need to have the best quality data before we start performing difference calculations. Now I saw a large number here, large in terms of what I can manage myself, abstract factors that I might want to try and use to analyze these data. Too many, four, 5. We're talking 5 dimensions in which we're working, and I would rather work in 2 dimensions. So I need to reconsider which part of this experiment I'm going to apply the techniques that I'm about to illustrate. So the idea is to find a part of this experiment where the variation is occurring, but it's only occurring by 2 spectra combining to form these variations. In other words, a 2-dimensional space. And I might guess that the end of this experiment, where the oxide has formed to a greater extent, that this might be representative of spectral shapes that actually sit in a 2-dimensional space. And we can test that hypothesis by applying the PCA calculation to a subset of these data. So when I perform a PCA on these data and look at the abstract factors again, that third abstract factor now looks like noise. So that suggests that I can reproduce all of these data in this part of the experiment using 2 abstract factors. I could return the data back to the state before I did the PCA calculation, but actually these buttons will reset the data and then calculate the abstract factors again and reproduce the data in one step. So I don't need to reset the processing. I simply press the AS2 button, and that will reproduce these data using 2 abstract factors. So I've now got improved signal to noise in a set of spectra for which I believe they all lie in the same 2-dimensional plane. These data are now all arranged in a column, overlaid according to the colours we see here. We're only interested in looking at the difference between 2 spectra, but when we organize the data in this format, the difference option will take the first and the last in the sequence, so that I can now construct another VAMAS file that will then illustrate visually what these different spectra look like and allow me to inspect and then determine which ones I prefer as potential component spectra for understanding the changes that we see within these spectra here. So let's apply the difference button, and here is the file that contains the different spectra. The first one is the top spectrum in that selection, and the bottom one is the last in that selection. So if I go back to the original file, you can see here this one is the first and this one is the last. They appear here in the interpolated file right here, bottom and top of this list. Every other one of these VAMAS blocks contains the first one minus a bit of the last one, and if I keep on scanning down this list, we will observe that these spectra then start to change. So you can see from the intensity scale that things are changing, and we can observe the spectroscopic shapes, and we reach a point where you say, well, that is no longer physically meaningful to have a negative going peak. So we would think that if the oxygen is non-physical, we ought to think that the titanium-2p is also non-physical. So let's go back up and see if we can find something that looks a little bit more plausible as an oxygen peak, and therefore we have an amount of oxygen that we can correlate with an amount of titanium, and the shapes that we observe are compatible with what we know from other samples containing titanium and oxygen. So let's take a copy of that. I've used this toolbar button. This is a toolbar button that simply copies the selected VAMAS block, and it shows you which one was selected. It also automatically selects the process data only, therefore it will always copy the process data. That's not an issue here, they're raw and process data are one and the same. And what it will also do is add an index to the element transition field so that the new VAMAS block will appear in a new column. And there it is. So if we scan down and look for that VAMAS block, it's still selected. If I use the arrow key and step left, I return to the column of different spectra, and I can carry on down my list looking for a potential alternative shape to the data. And there we have something that looks plausible. And the reason I believe this to be a plausible spectroscopic component is because whenever I've seen other experiments involving titanium, for example a multi-layer silicon dioxide titanium dioxide material sputtered, that I've seen this type of shape appearing within the titanium. So this shape is certainly a plausible spectroscopic shape for a form of titanium that has been damaged through the use of an ion beam and is relating to an amount of oxygen. So I'm going to take another copy. Therefore I have two component spectra that I have identified from the subset of spectra in the file. The final step is to now test these component spectra against the data that I previously used to construct these spectra. So let's take these. They're already selected, I believe. Yes, they're already selected. I'm going to go back to the data file. I have a selection here that is not active but all displayed here, so I'm holding the control key down and clicking twice will then select the VAMAS blocks that are displayed in the active tile. We also auto-verify that the two VAMAS blocks that were previously selected are still selected, and then I'll create a new file and copy all to the new file. So I've now got two component spectra. These are the ones that I've calculated. These are the raw data. I didn't try to take any PCA enhanced data. I just copied without the process data, so this is the raw data. These were already raw data in the sense that no processing was performed on the different spectra. So when I overlay these component spectra here, I can then compare these two spectra in different proportions that are calculated in a least square sense to the spectra that I have in this column that's selected using the linear analysis button. So I press the button and a new file will be created that contains the data, the two component spectra scaled, and the sum of the two component spectra in the least square sense so that they match these individual van mass blocks. So I'm going to perform that calculation, and we see the new file. There's the original data, the least squares, that's the sum of the different component spectra, two component spectra summed to give the least square solution in the correct proportions. So when I display these overlaid, you can now see that the data, it's probably easier to see if I zoom in, that red trace is the least square solution and the blue trace is the original data, and these are the two component spectra in the correct proportions to produce this red trace. So let's have a look at this again in more detail, and we will then use the arrow key to step down the selection, and we can observe how the data change over that interval of the profile, rather the oxidation sequence. So here we can see that we have a plausible answer in terms of we've got some titanium that is clearly related to oxygen, that it has a much more metallic-like look to it, so a reduced oxidation state here. We then have another state of the titanium that is correlated with a different amount of oxygen, and these two component spectra are enough to understand what's happening in terms of these two different phases as described by these component spectra. Now this is not the end of the story, this is just the beginning, because what we would like to do is carry on analysing the rest of the data set, finding other shapes that are plausible. We certainly have a metallic form that is very plausible because it's our initial state, so that ought to be included in the calculation. So we keep adding spectral shapes to the calculation, performing the same sort of analysis that we see here until we end up with a set of component spectra that give us some insight into what's happening as this titanium oxidizes.